Hello friends, today we'll be studying about the process of replication in DNA. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a copying mechanism for the genetic material. This copying mechanism is actually called or termed as replication of the genetic material, which is the DNA. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental DNA and one newly synthesized strand. This scheme was termed as semiconservative DNA replication. This semiconservative nature we'll discuss about it later. Before that, you should know what is replication and how is DNA replicated inside a cell. So replication, as you already know, the term itself indicates replication means forming a replica or exact copy of the existing strand of DNA. So the steps of replication in DNA: first is unwinding or uncoiling of DNA molecule. First, the DNA has to be unzipped, that is, the parental DNA molecule has to be unzipped, which is catalyzed by the enzyme DNA helicase, and this particular process occurs at a particular site called the origin of replication site. The tension in the DNA strand caused due to continuous uncoiling is released by topoisomerase. The single strands thus formed are stabilized and kept intact by single-stranded binding proteins. This means that when the double DNA helix uncoils, there is tension created, that is, more and more uncoiling in the front end or in the back end. This tension is released by the enzyme topoisomerase. Next, to prevent further coiling of DNA helix, it has to be kept intact, and this is done by single-stranded binding protein. Next is synthesis of nucleotide. The next step after uncoiling is that nucleotide has to be added to parental DNA in a stretch to form a newly synthesized strand. The small opening of DNA helix where replication occurs is called the replication fork. A short stretch of nucleotide called RNA primer is synthesized at the 5' end of the strand. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3'. You have to keep in mind very thoroughly that it is only in one direction, which is 5' to 3', that all DNA replication process happens and not otherwise around. This creates some additional complication in the replication fork. And what is that additional complication? You can see from this diagram that once this replication fork has started, you already know that the polymerization starts only at 5' end. So this is the area. One is this end and the other is this end where the replication starts, and addition of nucleotide happens, that is, polymerization of nucleotide happens. So in this strand there is continuously open stretch through which nucleotides can easily join in and form a newly synthesized strand. But on the other strand, there is only one short stretch available at a moment. So multiple RNA primers has to be synthesized for polymerization to take place. Hence, this leads to discontinuous synthesis, leading to the formation of fragments. This particular fragments, which is formed on the strand with polarity 5' to 3', is called Okazaki fragments. The step 3 is formation of two strands. The complexity which we were talking about in the previous one, that is this particular rule that DNA-dependent DNA polymerase catalyze polymerization only in one direction, that is 5' to 3', create complication. This complication is nothing but discontinuous synthesis of strand on template with polarity 5' to 3'. So this is the complication, and what happens is when these fragments are formed, these fragments are joined by the enzyme DNA ligase. So now after joining of the enzymes, we have two strands. This one is called the leading strand, which is the continuously synthesized strand, and the other is called the lagging strand, which is discontinuously synthesized. These fragments are joined by the enzyme DNA ligase. We have to keep in mind very thoroughly. So this here is the summary of DNA replication, the 5 steps. First is unwinding of DNA helix by helicase, then keeping the single stranded intact by single stranded binding proteins, the formation of the leading and lagging strands. The leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. The lagging strand is synthesized discontinuously. Primary synthesizes a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. After the RNA primer is replaced by DNA, it falls off and the DNA ligase joins the Okazaki fragments to the growing. Hence, the 2 strands newly synthesized forms. So after completing the DNA replication, you have got an idea how the DNA replicates. Now coming to the 3 models of DNA replication which were earlier proposed. After Watson and Crick proposed the double helix model of DNA, 3 models for DNA replication were proposed. And what are those 3 models? Conservative model, semi-conservative model, and dispersive model. Conservative model. In this model, the 2 parental DNA strands are back together after replication has occurred. That is, one daughter molecule contains both parental DNA strands and the other daughter molecule contains DNA strands of all newly synthesized material. So this, in short, from the diagram you can see that if this DNA double helix is getting synthesized or getting replicated, the 2 daughter DNA double helix which is formed is completely different. That is, one daughter is completely of the parental strands and the other daughter molecule is completely of the newly synthesized strands. Now coming to semi-conservative model, which is the accepted model. In this model, the 2 parental DNA strands separate and each of those strands serve as a template for the synthesis of a new strand of DNA, which you recently learned. The result is that 2 DNA double helices, both of which consist of one parental DNA and one new strand. So this suggests, semi-conservative model suggests that after replication, one daughter molecule has one parental DNA strand and one newly synthesized strand. Both the daughter molecules has one parental DNA strand and one newly synthesized strand. Now coming to dispersive model. In this model, the parental double helix is broken into double-stranded DNA segments, that is, as for the conservative model, act as templates for the synthesis of new double helix molecules. Then these segments reassemble into complete DNA double